Thank you for inviting the DecodeME project to present an update to you today. My name is Claire Dransfield. I work for Action for ME and we are partners on the DecodeME study. And also presenting is Professor Chris Ponting. He's the principal investigator on the DecodeME study and works at the Human Genetics Unit at the University of Edinburgh. DecodeME is a co-production and Chris and I are here representing the entire project team. The project's funded by the MRC and the National Institute for Health Research. DecodeME has two core objectives. Firstly, to identify DNA variants and genes that may influence a person's chance of being diagnosed with ME. So essentially, we're a genetic study. We've collected DNA samples from over 18,000 participants, which makes us around 10 times larger than any previous genetic study into ME. Our intention was always to create the biggest cohort in the world of people diagnosed with ME, and this will be an open source resource available for future research projects. And Chris will speak a little more to this in just a moment. So the genetic study is a GWAS, a genome-wide association study. This involves observing a genome-wide set of variants associated with a trait. In our case, we're comparing our ME participants with UK Biobank matched participants unaffected by ME. In order to recruit our participants, we developed a website through which participants could sign up. They could also sign up by phone, by email, or by post. Anyone meeting the eligibility criteria were encouraged to complete a questionnaire, and this was completed either online or by post. And we also elicited the help of the 25% ME group, who provided telephone support to those who may be more severely affected. The eligibility criteria were assessed through the DecodeME questionnaire, and cases were invited to provide a DNA sample. Participants' DNA was collected through a postal saliva kit. This made it as accessible as possible for all, including the more severely affected. Back in 1995, Woodward et al. Reported that obtaining a diagnosis was the single most helpful event for 90% of patients affected by ME or CFS. This allowed them to be less anxious or fearful, to formulate ways of coping with their symptoms, and to communicate with healthcare professionals and others more effectively. DecodeME is more than just a genetic study. We wish to play our part in reducing the stigma and raising awareness for those living with ME, to help to improve the relationship between people living with ME and health professionals, and also to provide a feeling of ownership and representation for the community. And on this slide, here are just some of the quotes provided to us when we ran a social media campaign in the middle of our participation recruitment, explaining why the DecodeME study meant so much to them. So this provides you an update on the project now we've finished the recruitment phase. We've achieved almost 38,000 sign-ups, and nearly 27,000 people completed our first questionnaire and consented to participate in the study. Over 21,500 people were invited to provide DNA, and of those, over 18,000 provided a DNA spit kit return to us. In January to March this year, we ran a second questionnaire, and a huge 15,000 people provided responses to that. Overall, 86% of DecodeME participants have consented to sharing their de-identified data with other researchers, and a huge 95% have consented to being recontacted for new research projects. This is really important in terms of building that cohort that'll be available for future researchers. And I'll hand you over to Chris, who will explain a little more about that now.
Thanks, Claire. I'd like to talk now about how we're providing researchers with access to DecodeME data. So this is only for data relating to people who have consented to us doing so. It's not for everyone. And our clear aim is that DecodeME is catalyzing new research opportunities for the future. So to be eligible for the data, the research needs to be feasible and with scientific value, and researchers will need to complete with data security and ethics arrangements. They will need to publish results. They won't need to publish with us. We're not requiring others to name us as collaborators, only to acknowledge that DecodeME data, your data, was used and where the funding came from. We want to remove as many hurdles from researchers as possible. We've piloted data sharing in 2 projects. The first on the left here is with Professor Ruth Jarrett from the University of Glasgow. And Ruth is studying herpes virus 6, but only copies of the virus that have been integrated into human genomes a long time ago and have since been inherited down the generations. The second on the right is with Precision Life, and we want Precision Life to test whether the gene combinations that they predict as relevant to ME/CFS are found not just once, but several times in different data, different types, different subsets of DecodeME genetic data. Now, we also want to replicate our findings. So we forged a new collaboration with Hanna Alila and Anina Turvi from FinGen, who are in Finland. So they're looking for genetic risk factors for ME as we are. So if they find something, we can replicate it or seek to replicate it. And if we find something, they can also seek to replicate it. I know we've said this many times, but it really bears repeating. Patient and public involvement, PPI, is at the heart of everything that we do in DecodeME. Every person, irrespective of whether they are a researcher or someone with lived experience of ME, participates equally in every decision. So this is, we believe, has instilled a lot of trust and also definitely has enhanced the science. For example, because the questions we ask the community are better framed and are more often understood by more people. So, of course, this project, DecodeME, is all about genetics. We do expect to discover a few, maybe up to 5 genetic risk factors of ME. So why is that? Because previous research, and in this slide, it's from Albright et al. In 2011. Previous research showed that people are twice as likely to have ME if they also have a first, second, or even third degree relative who have ME. So when we surveyed the first 17,000 DecodeME participants, what was really clear was that the symptom that most frequently was reported by them was post-exertional malaise. Here in this diagram, each data point is a symptom, and the further from the center of the circle it lies, the more often the symptom was reported by DecodeME participants. The second most frequently reported symptom was unrefreshing sleep, and then brain fog, and then fatigue. Now, females with ME, especially those who are younger, tended to report more and more diverse symptoms. So this data here are from the same set of 17,000 participants. And if you look closely, the only symptom that was significantly male biased was feeling easily annoyed or irritable. All the other ones that were significant were female biased. So not just in the numbers of females that are affected and diagnosed with ME that is substantially biased, but also the symptom reporting is also female biased. So here are the questionnaire responses or symptoms that significantly associate with ME severity. So at the top down, second one down, the older age is associated with greater severity. And at the top, disabling fatigue. So those are just examples. So returning to PPI, our PPI members, DecodeME PPI members, improved the wording of the questionnaire to clarify, for example, that PEM induces worsening of many symptoms, not just a fatigue, as indicated in this BACME 2021 document. And we feel that that's an important aspect that needs to be considered. So we're comparing our questionnaire results in DecodeME with the recently reported EMEA results from across Europe from 11,000 respondents. And what the EMEA showed was that pacing has a positive effect on people's ME. On the other hand, cognitive therapy as a coping tool had, on average, a very slight positive impact, a very tiny positive impact. Respondents tended to experience a strong negative impact of cognitive behavioral therapy as a cure for ME, and an even stronger negative effect for physical or mental activity that leads to PEM. So, what do DecodeME participants say about what works for them? So in the BACME primary care guide, it says that complete rest will not make ME better. However, when DecodeME participants were asked the general question about what therapies they had tried and whether they made them worse or better, here's what they told us. Most people said that pacing makes them feel somewhat or much better. Now, please note that this is a preliminary finding as we haven't completed our analyses, but you'll see that very few people said pacing made them feel much worse or somewhat worse. And similarly to the EMEA survey on the left, most DecodeME participants, that's on the right, report CBT to be ineffective. And roughly equal numbers report benefit or worse symptoms. So note the numbers here. Over 2,000 participants volunteered their experience for DecodeME. Again, this is preliminary. And finally, most DecodeME participants reported graded exercise therapy to be harmful. I'm showing responses containing either the word graded at the top or GET on the bottom. And this agrees with the revised NICE guidelines, which said that GET was harmful and should not be used, and that CBT is not a curative treatment. And of course, it also agrees with the EMEA questionnaire results. So thank you to everyone involved in DecodeME, from researchers to PPI to the thousands of participants. Thank you.